Hello, I'm Madison Gamble, and for the Enzyme Lab experiment we chose salt concentration. So enzymes are biological catalysts. They are composed of protein of three-dimensional shape which is affected by the amino acids. Enzymes have active sites where a substrate binds to and carries out specific reactions. Enzymes speed up chemical reactions by lowering their activation energy by using a catalyst. Lowering the activation energy allows the enzymes to use less energy and work faster. Enzymes are important for all living organisms because they allow them to respond to their environment and regulate their chemical reactions. The lab is mainly about trypsin, an enzyme which is located in the small intestines and is altered by different environmental factors, pH, temperature, and salt concentration. In our experiment, we focused on how salt concentration is affected by enzyme activity. Enzymes are involved in almost all reactions in living organisms, which also synthesize proteins, break them down, and speed up chemical reactions by lowering the enzyme's activation energy. These rules are very important to all living organisms, and if disturbed, can affect how organisms function. The role of trypsin in animals is to digest proteins in the small intestines. And for bacteria, the trypsin's activity is mainly found in the surface of the bacteria and breaks about the peptides from undergoing hydrolysis of larger proteins. The question we wanted to test for our experiment was, how does salt concentration affect enzyme activity? Our hypothesis was, as we increase salt concentration activity, enzyme activity will decrease as well as absorbance levels become of salts. Concentration will be able to change the charges of enzymes by impacting the hydrogen ionic bonds. So next, after that, we have our material methods. Here. TCA was added to stop enzyme activity. We centrifuged the reaction after adding TCA because we wanted to separate the solids from the liquids. The pellet contained at the bottom of the undigested azocasein, and the supernatant contained digested azocasein. The substrate used was azocasein in addition to the sulfanilamide. It is correlated to the measurement of the absorbance at 440 nanometers. This showed whether or not the enzyme was functional. For our control variables, the pH was 8 and the temperature was 45 degrees Celsius. We chose pH of 8 because it was the only one available, but our classmates also stated that 9. pH 9 was also the highest, but we could not do that for our experiment. And we also chose 45 degrees Celsius because the trypsin was more active in higher temperatures. For the negative controls, instead of adding enzyme trypsin, we added distilled water because we were trying to compare it to the test results since there are no outcome for the negative control. We did 2 sets for each of the individual trial. We did 4 replicates in order of the rule outliers and get more accurate results. So our first step was prep and label microcentrifuge tubes. Step number 2 is gather 3 microliters of each salt concentration. Step 3, pipette 450 microliters of the reaction buffer into each tube. Step 4, add 500 microliters of azocasein. 5, add 50 microliters of trypsin into the tubes and then place them on water bath at 45 degrees Celsius for 20 minutes. Tubes 5 and 6, also just a statement, were negative controls for each of the concentrations, so substitute trypsin with distilled water. Step number 6, once the time is completed, take the tubes out and add 500 microliters of 10% TCA. Step 7, place each tube in the centrifuge for 2 minutes at 12,000 RPM. Step 8, blend the center spectrophotometer at 440 nanometers using the cuvette with distilled water. Step number 9, transfer 1,000 microliters of supernatant to a clean cuvette. And our last step, which is step number 10, collect the data from the spectrometer and write them down results. Record each test under the 440 nanometer absorbance. So then as a little rewrap we did, so we first put the 450 microliters of each salt concentration into the 4 in their own tubes of 4 replicates of each. We chose 4 replicates to get more accurate results, eliminate outliers, as well as human error. Each concentration had a pH of 8, which was worked in the concentration. As I stated before, we don't have that available. pH 9, so we stick with pH 8, and we also increased to 45 degrees Celsius because that's where enzyme activity works the best. And then we added 500 microliters of azocasein, which is the substrate used in our experiment, to each of the tubes. Azocasein is modified milk protein casein because the milk protein with sulfanilamide added to be able to show us absorbance at 440 nanometers at the end of the experiment. Trypsin is supposed to be able to break down this protein, but with our reaction buffer salt, its effectiveness at breaking down the protein should be affected. After reading the azocasein, we added 50 microliters of trypsin to each salt concentration at the 4 replicate tubes and put 50 microliters of water into 2 negative control tubes. After adding a reaction buffer, substrate, and enzyme, we put all the tubes into a water bath heat at 45 degrees Celsius for 20 minutes. We chose the temperature based off of the other group's experiments. And after incubating, we added 500 microliters of TCA to the mixture to denature all the enzymes in the mixture so they'd stop working in the chemical reactions. After adding TCA, we centrifuged each of the tubes for 2 minutes to pellet all the undigested proteins from supernatant. Once we centrifuged, we were able to take the supernatant out and transfer it into a cuvette, and it reads as absorbance. And then for our results, this graph depicts the effects of salt concentration of the functionality of the enzyme trypsin. Here are my results charts right here. That trypsin became less active when salt concentration increased, which created the bars above clearly show the downward linear trend of enzyme activity in solutions with a high salt concentration. Since 0.15 moles of salt was our highest value of the graph, we chose to perform our t-test to compare it to other values. So 0.15 moles salt concentration compared to 0 moles, 0.75 moles, 0.3 moles, and 0.6 moles was found to not be significantly different because the p-value was greater than 0.08. When compared to other values, 1.4, 2.4, 4.8 moles of salt, comparing to 0.15 moles of salt was found to be significantly different because the p-value was less than the 7.94 to the 10 to the negative 6 power. Trypsin's functionality at 0, 0.07, 0.15, 1.2, 2.4, 4.8 salt had a standard deviation less than 0.03. However, at 0.3 moles of salt, the standard deviation was found to be 0.054, and at 0.6 moles of salt, the standard deviation was at 0.107. And then scrolling down here, we have our t-tests. Our t-test of 0.15 moles of salt compared with our first 4 values. We have 0.00 to 0.6 moles of salt was not to be significantly different, but the t-test of 0.15 moles of salt compared with the remaining values, 1.2 to 4.8 of salt, which we have here, was found to be significantly different. The standard deviation among the replicates was fairly low, with the exception of 0.3 moles of salt and 0.6 moles of salt, which were both equal to or above 0.05. The graph technically was still negatively linear once standard deviation was taken into account. So now we have here our conclusion. The experiment design was adequate in testing our hypothesis because we tested every available salt concentration, as well as 4 replicates for each concentration and 2 negative controls for every concentration. When composing data, no other group in our experimental did salt concentration, so there's no one to compare to. But when reading articles, we did learn through Greeners, finding that salt can affect the charges of enzymes by impacting their bonds. Therefore, when more salt is added, they should be able to break more bonds, making trypsin dysfunctional and ineffective to the azocasein. Potential errors that could cause variability after pipetting errors, incubation timing errors, as well as outliers. If we were to do this experiment again, we would most likely use more replicates to further prevent the number of outliers. In doing this experiment again, we would also like to test salt concentration close to 0.3 moles because without optimal temperature in our general experiment, we would like to test the concentration to get a more accurate optimal level of salt concentration for enzyme activity. Based off of our results, our hypothesis was correct that the higher the concentration, the lower the enzyme activity, thus a lower absorbance. Although our hypothesis was correct, we got a different graph than expected of getting a linear graph starting the graph with nonlinear, but ending up as the graph goes down. So it's kind of like a little bump in a way. At the lower salt concentration of 0.0 mole salt, the absorbance was 22.2 nanometers, while the highest concentration was at 4.8 moles of salt. The absorbance was at 69.3 nanometers. When comparing this data with enzyme to the negative control, we can see that when we replaced the trypsin with water, there was no activity, thus giving a high absorbance. For example, the negative control that had 0 moles of salt had an absorbance of 97.67 nanometers. For other variables that we use to help out with the different concentration of salt, we base them off of the experimental groups. These groups were testing the effects of temperature, had concluded that the highest enzyme activity was to be heated at 45 degrees Celsius, thus including us to make the same change to get the optimal enzyme activity. Groups not testing temperature were testing pH and found that the optimal pH for enzyme activity was 9, but we could not do that because of limited resources. Making sure that our variables and other salt concentration cause optimal enzymes activities was critical, and testing our buffer because we needed to make sure that the enzyme was working at its best condition in order to see how salt concentration is actually affecting the enzyme. In conclusion, we have discovered that The best possible conditions for enzyme activity in order to have the most optimal enzyme activity or grow bacteria should be kept in a warm environment at 45 degrees Celsius and a slight basic pH of 9 with salt concentration around 0.3 molars of salt. And that is our conclusion. Thank you.